Now we shall study about the prokaryote kingdom, that is life without a center. No doubt this lecture is getting longer, but today we shall cover first chapter completely. And in the last, we'll also have a question and answer session. So stay tuned with this lecture. And in the last, we shall uh, check and assess the ability that how much we have learned about living organisms. So imagine a study without a town hall or a computer without a central processing unit. Then that is the word of prokaryotes. This name literally means before the nucleus. If you remember the previous lecture where I have completely provided you information about prokaryotes. Okay, you can also re-watch that video. Okay, so the name clearly tells us that it means before the nucleus, while every other kingdom we have studied keeps its DNA locked in a safe. Prokaryotes, what does that do? They let their genetic code float freely in the open plan of the cell. So this kingdom is the foundation of all life on earth, and it primarily consists of the big two, that is bacteria, which are the most diverse group found everywhere from your gut to the bottom of the ocean. And secondly, cyanobacteria, which are also known as blue-green algae. These are the organisms that originally put oxygen into our atmosphere billions of years ago. And if you talk about the size factor, so to give you some perspective, these organisms are truly microscopic. You could fit about 1000 bacteria inside a single human skin cell. Okay, they are so small that they do not need complex transport system. They can move everything they need through simple diffusion process. So in the exam, here is a tip for you that sometimes you get confused about cyanobacteria plants. Okay, so if students often call cyanobacteria as a plants because they photosynthesize, you should be very clear about that. Even though cyanobacteria make their own food, they lack a nucleus. Okay, that is a major difference. They lack a nucleus and they lack a chloroplast. So if it does not have a membrane bound control center, that is a nucleus, then it belongs to the kingdom prokaryotes. Now we shall talk about study about anatomy of a bacterium. So think of a bacterial cell as a high-tech minimalist studio apartment. There are no separate rooms, there is no nucleus, there is no mitochondria, but everything is exactly where it needs to be for maximum efficiency. From propellers to sticky coats, let's look at the gear that makes them so successful. The first one is the external layers, that is the armor and the engine. We also call that as peptidoglycan cell wall, which is very critical point. Unlike plants which have cellulose cell wall or fungi that have chitin cell wall, bacteria use a complex mixture of proteins and sugars called peptidoglycan. It is incredibly tough and maintains the cell's shape. Second one is the slime capsule. This is an optional extra sticky layer. It acts like a space suit that prevents the cell from drying out and helps it stick to surfaces like the plaque on your teeth. And third one is flagella. A singular is flagellum. Flagella are not just tails that wag. They are biological motors. They actually rotate like a boat propellers to move the bacterium through fluids. And second one is the internal open plan. Okay, and where we have nucleoid. Since there is no safe nuclear membrane, okay, the DNA exists as a single tangled loop sitting directly in the cytoplasm. Here you can find that, okay? This coil. And then is plasmids. These are tiny extra circles of DNA. Think of them as cheat codes or superpowers. They often carry the instructions for antibiotic resistance, and bacteria can actually swap these with each other. And thirdly, 70S ribosome, that is their size. Okay, bacteria have protein factories just like us, but these are smaller than ours. We have 80S size, but this bacteria they have 70S. This difference is actually how some antibiotics work. They clog the bacterial 70S factory without hurting our 80S ones. When drawing a bacterium in your exam, never draw a circle for the nucleus, okay? Because they do not have any nucleus. Draw a messy wiggle of DNA in the middle and label it the nucleoid. Also remember the cell wall material. Its cell wall material is peptidoglycan, neither cellulose nor chitin. This is peptidoglycan. Spelling it correctly is an instant way to show the examiner you know your kingdom. That is Prokaryota. Uh, this is a quick recap of different shapes of bacteria. That is that there can be spherical, there can be rod-like shape, and there can be spiral. If you find bacteria in spherical shape, then the scientific term is cocci. Okay, if they are rod shape, then that is bacilli. Okay, and this is spiral shape. Okay, then those are spirochetes or spiral shape. Here are these three kinds of prokaryotes, okay, according to their shape. Secondly, some bacteria possess structures called flagella. This is flagella, okay. These are thin whip-like filaments that extend from the cell surface, okay. Flagella enable bacteria to move, they beat or flick, allowing the bacterium to protect itself through liquid environments. The next one is how bacteria reproduce. So imagine if you could clap your hands and suddenly there were two of you, identical in every way, including your memories and skills. This is the daily life of a bacterium. They do not deal with the complexity of finding a mate or the 10-step dance of mitosis. They use a high-speed copy and split method that is called as binary fission. The first step is DNA replication. Okay, so before this cell can divide, it must ensure the new cell has the instructions to survive. Okay, the single circular loop of DNA replicates, creating two identical copies. If the bacterium has plasmids, they usually replicate no two. The second one is elongation. Okay, the cell begins to grow longer as the cell membrane and wall expands. The two DNA loops are pulled toward opposite ends of the cell. This ensures that when the big split happens, each side has exactly one copy of the genome. And then is septum formation. That is a big pinch. Okay, like here, there's a pinch. A pinch begins to form in the middle of the elongated cell. The cell wall and membrane start to grow inward, creating a dividing barrier called a septum. And then the result, which are two identical clones. The septum fully closes and the two cells pop apart. Now you have two separate independent organisms. because there was no mixing of DNA with another parent. These daughter cells are genetically identical clones of the original. Sometimes examiners like to ask, why is binary fission considered as asexual reproduction? Your answer could be because it involves only one parent and produces genetically identical offspring. No gametes means no sperm, no eggs are involved, and there is no fusion of nuclei. Here's a quick recap of what we have learned about prokaryotes. They are unicellular organisms. They lack a true nucleus. They have circular DNA free in the cytoplasm. They possess cell walls made of complex materials. What is that material? Comment in the comment section. Okay. They may have slime capsule and flagella. They include bacteria and cyanobacteria. And despite their simple structure, prokaryotes are incredibly diverse and play essential roles in ecosystem, industry, and human health. Next, we shall read about Protoctist kingdom, which we also called as junk drawer of biology. Why? We shall come to know in this slide. So imagine you are cleaning your house, okay, and you find a bunch of items that do not belong in the kitchen, the bedroom, or the garage. What do? You put them in a junk drawer. Okay, so in biology, kingdom Protista is that drawer. It is a widely diverse group of organisms that have only one thing in common. They are all eukaryotic, but they do not fit anywhere else. Okay, then how to define the features of Protoctists? First of all, the nucleus, unlike the prokaryotes, means bacteria, we just studied, Protoctists have a control center. Their DNA is locked safely behind a nuclear membrane. And most of these are unicellular, means single-celled, which is why we often need a microscope to see them. However, there are giant exceptions like seaweed and kelp. Seaweed, if you remember, we just studied that in this lecture, in the start of this lecture. Okay, that how to differentiate between seaweed and seagrass. These are multicellular protoctists. Okay. So, secondly is diversity of nutrition. Because this kingdom is a junk drawer, the members eat in very different ways. We often categorize them by which major kingdom they act like. The first one is animal-like kingdom, that is protozoa. Okay, these are hunters. They move around and swallow other microbes like amoeba. Okay, which uses false feet, means pseudopodia to trap its prey. And plant-like, which we call as Protophyta. These are solar powered. They contain chloroplast and perform photosynthesis. Their example is Euglena. We shall study all these in our next slides. Okay, we are just moving to that. So if you look at a drop of pond water under a microscope, you will see tiny green specks zooming around. These are not just plants; they are Protophyta. They have the chloroplast of a tree, but the speed of a race car. These organisms are the primary producers of the underwater world. Without them, aquatic food chains would collapse. Two of the most famous examples are Chlamydomonas. Okay, Chlamydomonas, as you can see in this picture, the cup chloroplast. Okay, this tiny single-celled alga has a massive cup-shaped chloroplast that takes up most of its body. Secondly, it uses two long whip-like flagella at its front end to pull itself through the water toward the light. And then is Euglena. This is a biological hybrid. Okay, this is one of the most fascinating organisms in biology. When there is light, it uses its chloroplast to make food like a plant, means acts like an autotrophic. But If it gets dark, it does not starve. It does not starve, okay? It does not remain hungry. It can switch modes and absorb nutrients from the water like an animal. Means then that acts like a heterotrophic. And there is also an eye spot, okay? If it even has a red eye spot, that is also called a stigma, that it uses to sense where the brightest light is present, so it can swim toward it to process its food. Here I also want to give a exam tip that sometimes examiners love to use Euglena to test if you understand the difference between kingdoms. If they ask why it is not a true plant, what would be your answer? It lacks a cellulose cell wall. Okay, this is the perfect answer for this question. If you are asked about that why Euglena is not considered, is not included in the plant kingdom. It can feed heterotrophically like an animal, and it is unicellular and motile. It moves. So while these green microbes are busy making food from sunlight, there are other protoctists that are pure hunters. So let's look at the animal-like masters of the hunt, which are protozoa. If the plant-like protoctists are the grass of the pond, the protozoa are the lions and wolves. They lack cell walls and chloroplast, which mean they have the hunt and eat solid food just like we do. But since they are only one cell big, they have developed some of the strangest hunting tools in nature. First of all, we shall talk about amoeba. Okay, it does not have legs or fins. Instead, it flows. It pushes its cytoplasm forward to create false feet called pseudopodia. And when it finds a bacterium, it does not just bite it, it surrounds it. The pseudopodia reach out like arms, circling the prey until it's trapped inside a food vacuole to be digested alive. And then paramecium. While the amoeba is slow and creeping, the paramecium is a high-speed swimmer. Its entire body is covered in thousands of tiny hair-like cilia. It uses these cilia like synchronized oars to zoom through the water and sweep food particles into its mouth, which is a specialized dip in its side called the oral groove. And the third one is Vorticella. This protozoan stays anchored to one spot by a long coiled stalk. If it gets scared, that stalk can snap back like a high tension spring. Okay, in this table you can easily differentiate between all these four: amoeba, paramecium, euglena, and chlamydomonas. Okay, here chlamydomonas and euglena, they belong to Protophyta, while amoeba and paramecium, they belong to Protozoa. Now here is a comparison. If in an exam you might be asked to identify these based on the movement, means differentiate between protozoa and protophyta, or differentiate between amoeba, paramecium, euglena, chlamydomonas. Simply keep in mind that amoeba has pseudopodia, paramecium has cilia, and euglena has flagella. Okay, for their movement, and their nutrition methods are also different. Amoeba and paramecium, means protozoa, they have holozoic mode of nutrition methods. By Euglena chlamydomonas, chlamydomonas that photosynthesize, but Euglena that acts both like a heterotrophic and an autotrophic according to its environment. Now the question is why this kingdom is very tricky. If it has chloroplasts, why it is not just a plant? The answer is usually complexity. Plants are multicellular with highly specialized tissues, means roots, leaves, and xylem, while Protoctists remain simple, usually just a single cell doing everything at once. Now we have come to a very important and very different section, that is viruses. We shall study structure, its classification debate, and how it reproduces. This is very interesting, and do not miss any minute, any second of this discussion. So if you leave a cat in the room, it breathes, moves, and eventually gets hungry. But if you leave a virus in a room, it does what it does? Absolutely nothing. It sits like a pebble. So viruses are the most controversial group in biology because they are not actually cells. They are biological zombies that only come to life when they hijack a host. But why? Because they miss they miss Mrs. Gran. Okay, Mrs. Gran, what is Mrs. Gran? That is MRSGREN. Remember this first lecture? Movement, respiration, sensitivity, growth, reproduction, excretion, nutrition. Okay, so to be considered alive, an organism usually has to pass the Mrs. Gran test. Okay, outside of a host cell, a virus fails almost every single one. They have no respiration, they have no growth, no excretion, and no nutrition. The only thing they can do is reproduce. But even then, they can't do it alone. They have to hack a living cell and force it to make copies of the virus instead. And those are not considered as cells. This is the number one mistake students make on exams. Okay, a virus is much simpler than a bacterium that does not have any cytoplasm, any ribosome, and any cell membrane. Okay, so if a question asks you why is a virus not considered a living organism, your answer should be very clear. It cannot carry out life processes, seven pillars of life, remember that. Okay, so without but yes, that can, but will in a living host. Okay, so it cannot carry out life processes without a host cell. This is the key marking point that examiners look for every year. If a bacterial cell is a fully equipped factory, a virus is just a single set of blueprints. It does not need a kitchen, a bathroom, or an engine because it plans to steal yours. Let's look at the three parts that make up this ultimate biological hacker. Number one is the genetic core. That is a blueprint or DNA or RNA. Okay. Unlike us, viruses do not always use double-stranded DNA. Some use RNA, like the flu virus or HIV. This core contains only one set of instructions: how to make more of me. Number two is capsid. That is the box. That is also as a protein armor. The genetic core is wrapped in a protective shell called a capsid. Here you can see, okay, capsid. Then is capsomeres. The capsid is not one solid piece. It is built from identical protein units called capsomeres. Think of them like Lego bricks snapped together to form a shield. Okay, and the third one is envelope. That is a disguise. Okay, or you can say that as sneak layer. Some viruses are enveloped. As they leave a host cell, they actually steal a piece of the host own cell membrane to wrap around themselves. But why do they this? This fatty outer layer helps the virus trick the next host cell into thinking the virus is a friendly part of the body, allowing to allowing it to slip inside undetected. Here is a very common exam question which is being asked for two ways a virus differ from a bacterium. The golden answer is here: viruses have a protein coat that is capsid instead of a cell wall. Number two, viruses contain either DNA or RNA, while bacteria have both. Number three, viruses lack cytoplasm and organelles like ribosomes. So, to wrap up our journey through the kingdom of life, we have to look at the ultimate comparison. On one side, we have the living cells like us and bacteria, plants, which are like fully functioning cities. Okay, on the other side, we have viruses, which are like a single locked USB drive. Let's see how they stack up in a head-to-head battle. First of all, they have a cell membrane, okay, cytoplasm and often a nucleus or organelles. Simple, just a genetic code, DNA or RNA inside a protein coat, which have viruses. Okay, then by memorizing this table, you can easily differentiate between viruses and the living cell. And if an examiner asks you why viruses are being separated from animals, plants, and even bacteria, your answer would be very clear. The next one is how viruses reproduce. So now we have the blueprint and we have the box, but how does this tiny package actually takes over a giant cell? Let's look at the 6 steps of a hijack. That is also called as the viral life cycle. Imagine a stranger walks into a car factory, throws out the original blueprints and replaces them with plans for a bicycle. The machines do not know any better. They just keep building what the blueprints say. That is exactly how a virus works. It does not reproduce like a cell. It hijacks a cell and turns it into a virus-making machine. The process follows a ruthless 4-step plan. Number one is the attachment. The virus is highly specific. It has proteins on its surface that act like a key. It searches for a host cell with a matching lock, that is a receptor, and sticks to it. Then the entry step. Once the door is open, the virus injects its genetic material, that is DNA or RNA, into the host. The empty protein shell is often left behind like a discarded wrapper. The next step is hijack, means replication and assembly. This is the hacker phase. The host cell's own ribosomes and enzymes are tricked into reading the viral code instead of making human or bacterial proteins. The cell starts manufacturing thousands of viral capsids and copying viral DNA. And the last one is release, which we also call name as lysis. Okay, then the cell becomes so packed with new viruses that it can no longer function. Eventually, the cell membrane bursts, which and this bursting process called as lysis, and then it releases a swarm of new viruses to go out and infect neighboring cells. In this diagram, you can easily imagine that how a virus replicates. Here is a favorite exam question: is why can't a virus reproduce outside a host cell? The answer is always about machinery. Viruses lack ribosomes to make protein codes. They also lack enzymes, which are to copy DNA. They rely entirely on the host organelles to do that work for them. Here is a quick comparison between virus and bacteria. Okay, the difference in between their size is very clear. Okay, so you can easily read out this, and we shall not go in depth because we have also uh, discussed many things about comparison. Now we shall have a very quick recap of this complete chapter, okay, of this complete 4 lectures. First one is master classification table, domain, kingdom, and viruses. Viruses, they are not in any domain. They are non-cellular. They do not have any nucleus, okay, and they are parasites. Second one is prokaryotes. It includes bacteria, archaea, okay. Their cell type is prokaryotic. They have cell wall that is made up of peptidoglycan, which is a special material. They have no nucleus. And thirdly is Protoctists. They belong to eukarya domain. Okay, their cell type is eukaryotic. Their cellular material varies. Why varies? Comment in the comment section. Okay, they have nucleus. Yes, they have nucleus. The nutrition method is both photosynthesis and ingestion. The examples are amoeba and euglena. Then the fungi group that also belongs to domain Eukarya. Okay, cell type is eukaryotic and the cell wall is made up of chitin that does have nucleus, and the nutrition method is both saprotrophic and parasitic. Next one is plants. They also belong to domain Eukarya. Okay, their cell wall is made up of cellulose. They have nucleus. Okay, and their nutrition method is only photosynthesis. That's why they are called as autotrophic. Ferns, maize, octrees, all plants. We can place them in this group. And the last one is animals. Okay, they also belong to domain Eukarya. Okay, but they do not have cell wall. They does have nucleus. Their nutrition method is holozoic. Key examples: insects, fish, and all mammals. Here is the history of classification evaluation. We shall only study the key figures. The first one is Aristotle. Okay, that was 384 before Christ. His major contribution was about the ladder of nature, and he categorized organisms based on their blood criteria. The next contribution is of herbalist. They introduced artificial classification system. The next one is John Ray. Okay, he introduced a natural classification system. He proposed that classification must be based on internal structures, means seeds or bones. That is the birth of modern botany. Next one is Carl Linnaeus. He introduced binomial nomenclature system. That is a major contribution. Okay, his core logic was every organism gets a unique two-part Latin names, genus name and species name. Then the contribution is of Whitaker. Okay, he introduced 5 kingdom system and he divided life into 5 major kingdom. Which are those major kingdoms? We have discussed that in thorough detail in our previous lectures. Next is the contribution of Carl Woese, who introduced 6 kingdom systems. And he discovered that archaebacteria, which is very ancient, are chemically different from eubacteria, which are very true. He separated kingdom Monera into 2 separate groups. Next is modern science, that is 3 domain system based on ribosomal RNA. 3 super kingdoms based on DNA and RNA sequence. Here is strategy for you to come forward and understand this complete chapter. Before the 1600s, okay, you should note out these points if anyone is missing. Before the 1600s, people classified plants by how they could be used. For example, plants that cure headaches versus plants you can eat. John Ray changed everything. He argued that we should classify organisms by their internal structure. He was the first one to notice the difference between monocots and dicots just by looking at their seeds. He moved biology away from what can this do for me to what is this organism actually made of. This was the birth of modern biological anatomy. Secondly is the naming revolution. Okay, Linnaeus revolution, universal passport. Okay, imagine a scientist in Pakistan, in India, or anywhere in the world, a scientist in Brazil, or a scientist in Norway, all trying to talk about a mountain lion. They would all use different local names and get completely confused. Carl Linnaeus solved this by giving every species a universal passport, the binomial system, by using Latin, which is a dead language. Okay, uh, being dead language, it does not change. So by using Latin, he ensured that Panthera leo means loin to every scientist on Earth, regardless of the language they speak. He organized life into the nested hierarchy we still use today: kingdom, phylum, class, order, family, genus, and species. The third one is genetic revolution, Wu's DNA code. For centuries, we classified things by how they looked. If it had fur, it was a mammal. If it had scales, it was a reptile. But in the late 1970s, Carl Woese started reading the molecular code inside cells, specifically ribosomal RNA. He discovered that some bacteria were so genetically different from others that they deserved their own category. This led to the creation of domains archaea, bacteria, and eukarya, which sit even higher than kingdoms. We shifted from looking at the shape of the book to reading the text inside the DNA. In your exams, you might be asked why a classification system change over time. The answer is always new technology. We went from the naked eye of genre to the microscope of Linnaeus to DNA sequencing of Carl Woese. As our glasses get better, our map of life gets more accurate. Here you should remember that examiners usually need to focus on the five kingdoms, but knowing the three domains is often required for the higher mark questions about modern classification DNA. So the three domain system is the most modern system because it relies on molecular evidence, that is RNA, whereas the older system relied mostly on morphology, that is their physical appearance. So congratulations, we have covered our chapter 1 that was in very detail, okay, and I am very sure that if you attended these all lectures of chapter 1 by attentively, okay, you have no need to consult any other lecture. You can easily understand everything from these lectures and from these slides. So here is the question answer. Okay, uh, no doubt this concludes the full lecture series on classification and the kingdoms and the domains of life. Now we are starting our question session. I should know that what you have learned, but I should assess that. So I am very hopeful you have learned much about this chapter, and now this is the time to check, to assess your ability, your knowledge, and competence. In next slides, you will have three types of questions. I'm not saying that those questions are from the previous books and from the previous exams, but definitely those are very comfortable to assess your knowledge about this chapter, okay, from this information. So in next slide, you will have three types of questions, including MCQs, fill in blanks, and then matching column. Okay. So by attempting these questions, you will check about your ability, and you will also assess your own ability, that how much have you known about living organisms and non-living things, how can you distinguish between them, how classification systems were evolved, and how biological classifications traveled according to timeline. These questions are covering the whole chapter, not only this lecture. Okay. If you know that we covered this chapter 1 in 4 lectures. So if you remember, the chapter 1 was covered in 4 lectures. So before attempting this question session, make sure you have gone through these lectures. Secondly, you will answer these questions in the comments section, mentioning the question number and the possible answer like 1-D. Okay, that is a supposed answer. Okay, you can write down that in comment section. So in matching columns exercise, you will follow the same method and will write down your answer in the comments section. For fill in blanks question, you will provide your answer according to the order. Those shall appear in the sorry for that, in the paragraph. So let's move to our questions. The first question is, that is MCQ. Okay, you will find 12 MCQs here. And which of the following is the correct way to write the scientific name for a line using binomial nomenclature? You have here 4 options. You will have to write down only one answer. One mean question number 1 dash, and then you will write down the answer. For example, I'm not saying that is the correct answer. Okay, I'm not saying the correct answer. You will tell me the correct answer, but I'm giving you just only the example. 1 dash A. Okay, and according to this you will write down your answers, and I shall ensure you the proper reply of your answers. Second question is, an organism is multicellular, has a cell wall made of chitin, and performs saprotrophic nutrition. Which kingdom does it belong to? Cell wall made of chitin and performs saprotrophic nutrition. So which kingdom does it belong to? Plants, animals, fungi, or prokaryotes? Number 3. Which scientist was the first to clearly distinguish between monocotyledons and dicotyledons based on internal structure? Aristotle, John Ray, Carl Linnaeus, or Robert Whittaker? Number 4. In the three-domain system, which domain includes all organisms that possess a membrane-bound nucleus? Eukarya, archaea, bacteria, or monera? The fifth question is, a virus is not considered a true living cell because it lacks which of the following? It lacks genetic material, a protein coat, the ability to reproduce, or cytoplasm and organelles. Question number 6. Which vertebrate class is characterized by having feathers, being homothermic, and laying hard-shelled eggs? Birds, reptiles, mammals, or amphibians? I think this is quite easy for you. Question number 7. A plant has leaves with parallel veins and floral parts arranged in multiples of 3. Which group does it belong to? Dicotyledons, ferns, monocotyledons, or mosses? Question number 8. What is the name of the thread-like structures that make up the body of a multicellular fungus? Cilia, hyphae, rhizomes, or flagella? Question number 9. The prokaryote kingdom is distinguished from all other kingdoms because its members lack DNA, ribosomes, cell wall, or a nuclear membrane. Question number 10. Which of the following describes the movement of an amoeba? Cilia, which are tiny hairs, flagella, that is whip-like tail, pseudopodia, that is the flowing cytoplasm, or contractile stalk. Question number 11. Which features are found in all prokaryotic cells? chloroplast and the nucleus, cell membrane and cytoplasm, mitochondria and ribosomes, nuclear membrane and DNA. Last question of MCQ section. Which group of arthropods has exactly three pairs of jointed legs and a body divided into three parts: head, thorax, and abdomen? Arachnids, crustaceans, insects, or myropods? You will answer all these MCQs in the comment section. Okay, students, the next one is matching the column activity. Okay, so here you will match column A with column B with its correct description. For example, if in column A1 is related to F, for example, I'm just giving an example, not a definite answer. Okay, answer is being expected from you in the comments section. Okay, if column A is corresponds to column B, 1 corresponds to F, then you will write down 1 dash F. Then I shall understand that how you're answering your questions. In the same way, the next one is fill in the blanks. In this paragraph, you will fill the blanks with the word bank giving the on the bottom of the slide. Here are few words, that is binomial, cellulose, prokaryotes, saprotrophic, chitin, eukarya, hyphae, capsid, heterotrophic, and genus. Okay, you will place all these words in the on the appropriate places in this paragraph, and then you will answer that in the comment section. For example, 1 prokaryotes, 9 genus. Okay, in this following this method, you will answer to this section. Here I shall talk about a few mistakes you commonly students make in their exams. So first of all, you confused about the cell walls, saying all cells have cell walls or that all cell walls are made of cellulose. No, this is not the correct answer. Remember, animals have no cell wall, plants have cell walls but made of cellulose, fungi have cell walls made of chitin, and bacteria have cell walls made up of peptidoglycan. The next mistake is writing scientific names incorrectly. For example, the mistake is capitalizing both names. For example, Homo and Sapiens. H and S are capital, or not underlining when handwriting. Okay, so to fix that issue, remember that only the genus, only the first letter of genus is capitalized, and always underline the name on your exam paper. Both names, genus name and species name, both names are being underlined. The third mistake is confusing in between antibiotics and viruses, suggesting antibiotics can cure cold or flu. Remember, cold and flu are viral disease. Okay, these are not bacterial disease, and antibiotics only work on bacteria. Why viruses do not work as antibiotics? Because they do not have cell walls or ribosomes for the drug to target. That is the reason that antibiotics do not work on viruses. The fourth mistake is misidentifying Monera. Using the term Monera in a three-domain question is wrong. Remember, Monera is an outdated kingdom. In modern exams, use Prokaryote for the kingdom and bacteria or archaea for the domains. The fifth one is uh, confusing between excretion and egestion, using both terms interchangeably. No, this is wrong. Excretion and egestion both are quite different terms. Excretion is getting rid of metabolic waste like carbon dioxide or urea, while egestion, that is getting rid of undigested food, for means feces. Sixth common mistake is binomial rank confusion. That is thinking that the first part of the name is the species. No, first part is the genus, always the genus. Think of it like a surname that comes first. Okay, that is just for your understanding. but remember that first name is genus and second name is species. That is very easy to understand. Seventh mistake is about ferns and seeds. For example, the mistake is classifying ferns as seed producing plants. No, that is wrong. No doubt. Ferns have roots and stem, but they reproduce via spores, not through seeds. The eighth common mistake is interchange homothermic and polyothermic. Okay, saying or simply saying that as cold-blooded or warm-blooded. You should use the technical terms, that is homeothermic or poikilothermic. Homeothermic means maintaining a constant internal temperature regardless of the environment. And the ninth mistake, common mistake, I'm not saying that you all will do that mistake, but this is just as a precaution for you. Ninth mistake might be confusing about nucleoid or and nucleus, stating that bacteria have a simple nucleus. No, bacteria have no nucleus. Remember, bacteria have no nucleus. That is a major difference. Okay, bacteria have a nucleoid region, or you can say that a circular loop of DNA. The tenth common mistake might be arthropod leg counting. That sometimes you are giving a question to identify either it is an insect, arachnid, or a meropod. Okay, you should first count the number of legs. So miscounting legs in diagrams can be a mistake. Remember, insects have 6, arachnids have 8, crustaceans have 10 plus. And always double check the image in the question to correctly answer and perfectly respond to that corresponding question. So I thank you all for joining today's lecture. Okay, it was a very long lecture, but I'm very hopeful. But I'm sure that you have learned many new things about chapter one. Okay, I conclude this lecture with this famous quote of Carl Sagan that science is a way of thinking much more than it is a body of knowledge. Okay, and at last, I just give you just a few reminders that do not forget to check the study guide for the 5 kingdom characteristics. Number one, number two, complete the MCQs, matching exercise and filling blanks to master your technical vocabulary. Okay, and answer all those questions in the comment box and prepare yourself for our next session, okay, where we shall dive into cell structure and organization. And do not forget to subscribe this channel. Okay, in this on this channel you will find all the lectures of biology, and in the future we shall cover the other books of physics, chemistry, and even mathematics. Okay, so stay tuned, stay subscribed, and see you in the next lecture. Till then, goodbye.